have green frame up there? We're good to go. Slideshow from the beginning. Okay, so cancers and tumor spread. We talked about the genetics, and what you're going to find one key theme with cancers is cancers always screwed up genetics. Not because your parents necessarily gave you those bad genes, but because something bad happened in the meantime, like a mutation happens. All right, here are the main objectives we want to get through today. And then first we're going to start talking about what cancer is. And if you watched the video, which hopefully you already did, then what did Gunther von Hagen say cancer meant in Greek? Crab. Yeah, because when they first started taking cancers out of people, they could see a tumor growing. And when that person would die and they pulled the cancer out or the tumor out, it looked like a crab because it had all these branching arms. It looked like it was invading and taking over. So they called it cancer. All right, another term you need to get familiar with is neoplasm. We talked about this before. This means new growth. Metaplasia or dysplasia, which one typically leads to cancer? Dysplasia, abnormal adaptation. It wasn't a true adaptation, it was abnormal. All right, and we're going to discuss the difference between benign and malignant tumors too. When you look at cancer, cancer is the second leading cause of death. It's still amazing. From the first week of class when we talked about what killed people 100 years ago was primarily some type of infection. Cancer really wasn't even at the top, way at the bottom of the list. Same with heart disease. Since we've fixed most of the infective agent problems, now we have heart disease and cancer as the top two killers, and they're pretty close to each other. It's a little over, I think it's right under 600,000 people a year are killed by heart disease, and then about 566,000 a year by cancer. So they're pretty close. And if we keep making progress in heart disease, we'll actually get it where cancer catches up to it. Cancer is so difficult because you've got that 3 billion base pairs of genetics in your genome, and all it takes is one change of base pairs to actually cause the cancer. So there's so many possible different causes, it's hard to actually distinguish one. So cancer is hard to cure. The benefit is that the overall mortality rate of cancer is 50% in 5 years. The primary reason is, anybody want to take a guess at what the number one type of cancer is? The most abundant cancer is skin cancer. Why is it not the number one killer? It's actually the number one treatable. Because you can see it, right? You have a cancer that's growing in your GI tract. Even if it's as dangerous as the skin cancer, you can't see it growing. And at the point where it starts causing symptoms, then that thing's usually huge or problematic. So skin cancer is the number one cancer, but they're actually the least fatal because they're the most curable. You get an ugly little bump on your skin, you freak out, go to the dermatologist, and they freeze the sucker off. The cancer doesn't have a chance to spread. Okay, so skin cancers. And then prognosis varies based on a lot of things, like your gender, your age, the site of the development. Like, we already know some of the bad cancers, like pancreas. Is that an okay cancer to get? It's terrible. It's one of the worst cancers because pancreatic cancers have one of the worst survival rates. The pancreas, when it's affected, affects so many different things at one time, and it's really hard to get by it. In fact, a lot of people will die from pancreatic cancer within a few weeks of being diagnosed. I had a friend, and it's been probably 7 years now. I was still at Iowa State when this happened, but he had been dating this person for almost 20 years. And they were dating, and finally they got married. And about 4 months after they got married, he got really sick the week before Christmas. They took him to the hospital. He went into a coma. When he came out, they told him he had pancreatic cancer and died 2 days later. So it was just amazing. Within, about a week and a half, he died of pancreatic cancer. All right, but some of the sites of development can be worse, like males and females with lung cancer. Lung cancer is bad, just bad in general, because those cells, they don't repair themselves when they've been damaged. Can you live without one lung? You can, yeah. So you're not going to be a marathon runner, most likely. I say that, and there's probably a marathon runner out there with one lung. But, yeah, it changes your lifestyle severely. When you look at some of the most dangerous cancers based on sex, which is the next one, you can see breast cancer and prostate cancer. My first semester teaching this, I actually had one person say, why is prostate cancer up in the female section? That's why I put the anatomy videos back on. But anyway, yeah, sometimes you want to recheck that anatomy. And then age. And of course, age is going to affect not just how it affects you with the cancer, but what type of cancer you get. I think it's the next slide. If you look, most of the carcinomas, the dangerous ones, usually happen in the 70s. A lot of the melanomas, like skin cancers, happen in their 20s and 40s. But you can see, based on your age, the type of cancer that affects you. So if we look at just like leukemia, who's it affect mostly? Older people or younger people? Leukemia is the younger people, yeah. More leukemias are happening to younger people. And you can see it kind of drops off in the middle years and then it starts coming back. And then if you look at something like breast cancer, you see breast cancer rapidly increasing. So after puberty, you'll see it fairly see it before puberty, and just keep rising and rising and rising. So, and then other cancers, miscellaneous other cancers like skin cancers up in here. So you can see cancer is age dependent too. Cancer, one of the things I want you to keep in mind is it's based on genetics, it's mutations, and it's really an accumulation of mutations. It's rare to get one mutation and that causes a flat-out cancer. It usually is mutation after mutation after mutation. We'll talk about that in a little bit soon. Okay, so how do we name the tumors? When you look at the first groups, the benign tumors, actually, what's the first thing you almost always see at the end of a tumor's name? Oma. It's not the Oma. Okay, sorry. Challenge Schwarzenegger there. But anyway, yeah, you see Oma at the end. And then you just have to be familiar with some of the prefixes. Some of them are easy, like lipo. What's that obviously telling you? Fat, lipids, yeah. So erythro would be based on a red blood cell. Yep, and then you throw an Oma on the end of it, and it tells you what the cancers are. Some of them are tricky, though. Because if you look at something like adenoma, it's talking about epithelial tissue, and it's talking specifically about epithelial glands. So an adenoma is specifically talking about epithelium and glands. What do you think a glioma is? Brain tissue, but not a neuron, because a neuron would be a neuroma, right? So a glioma is the supporting cells in the nervous tissue, where a neuron is the actual neuron itself. Why do you think it's rare to see true neuromas? Because neurons, when they're differentiated, they don't divide. So to make that neuron divide again, that mutation had to specifically affect its ability to replicate. And now it's differentiated, so it shut that off, so that cancer has to actually turn it back on. It's pretty rare to see real true neuromas. You see a lot of gliomas that we just refer to in general as brain cancer. Right? Leiomyoma is another tricky one. Leiomyoma is referring to smooth tissue of the uterus. So leiomyoma, smooth tissue of the uterus. And I'm not going to go through and list thousands of different names, but some of these you'll see. So leiomyoma is one you'll probably see down the road. Uterine cancer is kind of a common cancer. How about a chondroma? Chondra. Cartilage tumor. Yeah, so chondroma is a cartilage tumor. Where might you see that? Where do you usually hear? Like the joints, right? So bones and joints, chondromas. And I talked about lipomas. I started talking about epithelial tissue. Epithelial can be a little tricky because you have two names for different areas of epithelium. You have an adenoma, which we said was a gland, yep, and then you can have a papilloma, which is the actual epithelial sheet. So a papilloma is the actual sheet, where adenoma is the gland itself. And these are benign tumors. So just throwing that oma at the end of it. There are a couple types of tumors that they're never benign. Never, 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 never. So you never have a lymphoid tissue that's benign. If it's a lymphoid tissue, it's always considered malignant. And the same thing with white blood cells. You never have a benign white blood cell tumor. It's always a cancer. And what do we call those white blood cell cancers? Leukemias. So those are just some of the exceptions. Where would you find an have you name it? What would it be? A bone tumor? An osteoma. Yeah. Yep, just the plain oma refers to a tumor. If it's a malignant tumor, then they usually give them a special name, like a carcinoma or a sarcoma. So if you have an epithelial gland and it's an adenoma, it's a what? Malignant or benign tumor? Adenoma. It's benign, but when you call it an adenocarcinoma, it's a cancerous, it's malignant, that's dangerous. So there are a couple of words that you substitute and throw in there that distinguish between benign and malignant. So that sarco or the carca are the two main ones. So like adenocarcinoma would be a malignant tumor of the gland, so adenoma, it's benign. And then if it's connective tissue, then we refer to it as a sarco. So things like in the bone, or the connective tissue that supports the bone. So rhabdomyosarcoma is the connective tissue between the bone and the muscle. Rhabdomyosarcoma. So what do you think you would call just a benign version of that? Rhabdomyosarcoma. Just a rhabdomyoma. Yep, you drop the sarco out of it. And then I already mentioned this. So lymphatic tissue and blood-forming tissues, they're never benign. They're always considered malignant because they always travel. And they always get into the blood and they travel all over the place. Or get into the lymph tissue and circulate. So when you're describing these, some of the ways to distinguish between benign and malignant, one is the name. But the name's kind of the final step of labeling this thing. When you look at benign tumors, they have a tendency to grow slowly. They can grow fast, but they typically grow slowly. And this is kind of a guide instead of an absolute list. Where malignant tumors have a tendency to grow rapidly. So benign tumors like to grow slow, malignants like to grow rapidly. When you look at the capsulation, so I wish I had taken pictures, but when I worked in the lab, rats grow tumors a lot. So when I worked with rats, you'd have this rat and it has a big bump or a lump somewhere. And when we started doing surgeries on them, I got a little bit close to one time and I looked and it was just this lump of mass. And what I did is I took a scupula. If anybody know what a scupula is? It's that little thing that looks like a spoon for a lab. And I took it and I just slid it underneath and popped that off completely. I completely removed it with just a scupula. It wasn't sharp. It just popped off. It's because what had happened is it had a defined capsule. This tumor was growing. It was a rapid growth, but it had really well-defined tissue around it, so I could actually go underneath and just pop the whole thing out. And then it actually became kind of, it didn't really help the rats. It was just more of a nuisance for the rats by taking it out, but it actually became kind of fun if a rat had that would actually make that little incision. And put under anesthesia, of course, so it was all good, and then pull those things out. It was kind of a neat experience. But those were benign tumors. They weren't fatal to the rat. The only time you have to worry about benign tumors becoming dangerous is if they're stuck in an area that they can't expand. What part of your body do you think you could have a benign tumor in that would be life-threatening? In the brain, yeah, because you've got the cranium surrounding it. And it's trapping that tumor in there and it's smashing the brain. So benign tumors can be dangerous, but typically we don't associate them with being life-threatening. So well-defined means they have some kind of capsule around them. When you look at a malignant tumor, though, they branch out, like that tumor cell up in the top. It starts digging in and eating its surroundings and moves through different types of tissue. So it's hard. When you have a malignant tumor, that's why when they try and take the mass out, a lot of times little pieces are left over. With those rats, I could just slide under it with a scupula, pop the whole thing out, and I knew I'd isolated the tumor completely. Right, next thing, non-invasive. So like I was saying, with the benign, you have that well-defined capsule, which they call a stroma. And it's not invasive. It's isolated to that one region. So we can go in, pull it out, and not have to worry about it. With the malignant tumors, you have to worry about because they get in the blood or the lymphatics and they travel through the body. They can metastasize. Benigns are well differentiated. This is talking about the cells themselves. Those cells, if you pull out that tumor and I slice it open, look, most of those cells look like the tissue it was brought out of. Like if it was an epithelial tumor, they look like epithelial cells. If it was a connective tissue tumor, it looked like connective tissue cells. They were well differentiated. So if it were a benign neural tumor, and I pulled it out, it looked like a bunch of neurons right there. Right? Poorly differentiated, these things look like stem cells again. They've actually gone from a stage of almost being differentiated, and they've like regressed and gone back to stem cell capability. They look highly mitotic and like their stem cells rapidly replicating. So that's what it means poorly differentiated. If I pull this out, it's hard to tell where it came from because it looks like a stem cell, like a germ cell. Where a benign tumor, if I pull it out, it just looks like a lump of that specific cell.
The top cell there, is that one cell or is that multiple?
That one's actually one cell. Yeah, it's one cell isolated on a petri dish and then has an electron microscope. Yeah, I forgot to point that out. I think this is the same one as the first slide. So. And then low mitotic index, it means that they're going through mitosis, which means if it's low rate of mitosis, what would you say about the growth then of benign? It's slow. Yeah, so low mitotic index means it's going slowly through mitosis. High mitotic index, malignant tumors, these things are growing rapidly. If you extracted a bunch of the cells, you'd look at them, you'd see all the different phases of mitosis. So prophase, metaphase, anaphase, telophase, you'd see all those phases all over the place. These things are growing like crazy. What's interesting is that these cells, we always think of them as, this cancer growth as just destroying the healthy tissue, but they're actually destroying other cancer cells too. They don't care. They're like a pack of zombies or something. They'll eat anything and destroy anything in their way. So these are really dangerous cells, where these are isolated. And we'll talk about why these can grow so fast too later when we talk about blood vessel. And then the last one's with benign. They don't metastasize. They're isolated to the area where malignants can metastasize. And we'll talk about how certain cancers that develop in certain parts of your body, like metastasize to certain other areas of your body. Like breast cancer loves to move to the brain because the tissues are similar enough. So we'll talk about that. Cool, the arrows work. Right, next one. So carcinoma in situ. Some people pronounce it in situ, it's in situ. I always remember in situ because it's isolated to the site. So I always think of this as a transition from benign into malignant. You've got this normal tissue. What kind of tissue is this? What are the four types of tissue? Epithelial, connective, muscle, and nervous. And this is epithelial because what do you know about the pattern? Well, yeah, it looks like it's columnar because of the way the nuclei line up, but it's simple and columnar. You know it's epithelial because look, they're holding hands. All of those cells are connected. Nice, tight, sheet-like layers. So what would you call the stuff underneath it? Taking you way back to that anatomy. That's connective tissue. Yep. So up here you have epithelium. Down here you have the connective tissue that holds it in place. And then there's muscle, which is muscular tissue. And then the submucosa, which could be fats or any other kind of connective tissue. But with normal tissue, you see distinctive layers. With dysplasia, this cell starts becoming dysplastic. It starts getting this abnormal growth. It starts acting like it's a stem cell again. So it keeps replicating. Look what it's doing to those layers, making them all crazy. And then with in situ, it's still isolated to the epithelium. And then after it becomes in situ, it actually invades the other types of tissues. So in situ means it's still isolated to the epithelial layer. It's just an abnormal epithelial layer. This would be perfect if this were on the surface of your skin. That would be something like a mole or some kind of abnormal growth. We'd look at that and say, ugh, that's hideous. Freeze it off, and then you're safe. So they always freeze down or dig down a little deeper to make sure they got them all out. But in situ means it's isolated to that tissue. Right? So like I said, it's a transition between benign and malignant. The nice thing is that in situ cancers, they're 100% curable. Just freeze them off. Just cut them off. They usually dig down a little bit deeper. I was listening to Dr. Radio last week about a person that had one on their nose, and they're like, I don't want them to dig deeper because it'll leave a mark. Hello, you're going to live. Leave the damn mark and live. One place you'll hear about this in situ a lot is actually in ovarian or uterine cancers, or cervical cancers. Probably cervical cancer will be the most common one you'll hear about. Because when they're doing a pap smear and they see abnormal tissues, dysplastic tissues, then they go in and they actually take a larger chunk out and they can actually take the, ideally, they can remove the whole area of the inside of the cancer. So it's gone. So 100% curable, and typically we're going to hear them are basically on the female parts like the cervix. Okay, cancer cell characteristics, the next one. 10, do you like the way the tops are worded? Notice that all the slides I'm putting in the form of a question. So where can these cancer cells come from? That way if you wanted to use them as study notes, then you can just use each slide individually. I'm learning still. I'm adapting. So where can these cancer cells come from? 3 basic places. You can either have a normal stem cell. In a situation of like leukemia, a lot of times that's what happens. We'll talk about that when we get to childhood cancers. So in certain situations like leukemia, the stem cell was dysfunctional, and that stem cell just got out of control. It could be a cell that differentiated, like that neuron we talked about before. It became a neuron, it had a function, but some mutation happened to it, and now suddenly its ability to replicate came back on. Muscle cells would be the same idea. It's really hard to find a muscle cell tumor because muscles, once they differentiate, they don't want to replicate. How about smooth muscle then? Smooth muscle replicate? If you're a woman, I hope you say yes, because the lining of the uterus, the smooth muscle, is constantly replicating any other time of the month. So when you look at things like cardiac, you hear much about a heart cancer? Pretty rare. They don't differentiate. Right? And the third one is what we call progenitor cell. And the progenitor cell is kind of in the transition. It's going through the differentiation, but it hasn't completely differentiated yet. And then something happens to it, and it turns into a cancer. So if you think about the ages of these things, here's a baby cell, here's an adult cell, and here's your teen going through adolescence. So stem cell is the baby, the differentiated is the adult cell that's become what it's supposed to become, and the progenitor is somewhere in between. They can all mutate and become cancers. All right, so what three properties of transformation do we usually define cancer cells with? The first one's called autonomy. Autonomy means that they're independent. They don't act under the rules that govern all the cells in your body. Your cells have rules. They say, if you're meant to be a skin cell, you grow and you replicate through mitosis until you hit a wall of other skin cells. So if we take one skin cell and we put it on a petri dish, I guess I took that picture off, but if we took this petri dish here and you put one skin cell on it, the skin cell would replicate and replicate until it hit this wall. Then it would replicate to this wall, and this wall, and this wall. Once it hit the 4 walls, it would stop growing. So you'd actually have one sheet of skin across that petri dish, and it'd be happy. As long as you kept it giving it nutrients, it would stay happy as one sheet. The problem is that if this is a cancer cell, they're autonomous. They don't obey the rules anymore. They're kind of rebellious. So they hit the wall, and they say, screw this. You can't limit me, and they start growing upwards. So they start growing and growing and growing, and if they can't grow flat across the sheet, they'll start growing up the sheet, too. So you'll see this big, huge mound of cells. They're not governed by any of the rules of the cell. They're autonomous. And we call them anchorage independent. Normally when two cells bump into each other, they have these little ECAD heron things that bind to each other, and they say, hey, you're a neighbor, you're human, let's merge. And then they just connect to each other, and they glue themselves. They make those junctions that you heard of, these little tight junctions, and desmosomes. And they know to turn off. If this is skin and I cut the cell over here, now this one's not hooked to anything. What's it going to want to do? So if I destroy this cell, what's this one going to want to do? It wants to replicate and fill in that cell. When you get a cut in your skin, all those living cells are on the outside, start replicating, they start moving towards the middle, and then they fuse in the middle, reforming new skin. So it's a nice smooth sheet like this across here. Cancer cells don't do that. Cancer cells just keep abnormally growing, so they have autonomy. They don't abide by the rules. Next, they're anaplastic. Anaplastic means without form. They don't look like a normal cell. If you take a normal cell and you put it in the brain, it's the stem cell, it starts looking for like neurons or glial cells. It has a special form. If you put it on the skin, it starts looking for like skin cells. Anaplastic means it's hard to identify. So it may be meant to look nice and round like this one, but when you get done, it has this really weird abnormal shape. There's no specific differentiation. Cells that we normally refer to as anaplastic are placental cells. So placental cells or embryonic cells, stem cells. They don't know what they're going to be yet. You put them in a petri dish, you stimulate the environment and say, hey, this is a brain environment, and they start becoming brain tissue. If you stimulate them and say, hey, this is a cardiac environment, they become heart tissue. They become whatever they want, or whatever the environment they become. They differentiate and get a special shape. A cancer cell loses that distinctive shape. So it goes back and can actually change its form completely. We call that anaplastic, without shape. Number 3. So the last characteristic, self-renewal. And now, I can't reach that high, so I'm going to have to use the little hand up here. So a normal cell, we've talked about this before, as long as there's no death cue coming in, a normal cell is nice and healthy. If a death cue comes in, what does a normal cell do? What's the process it goes through? Apoptosis. Yeah, it commits apoptosis, shuts off, and you don't have to worry about it. It's a bad cell. There's some kind of cue that says, there's something wrong with you, kill yourself. And it does that. What happens with a cancer cell is it bypasses all those regular signals, like the death cue. If there's something wrong with the cell, and it bypasses that signal, now it's a cancer cell. There's survival cues. Usually it takes some kind of outside influence to tell the cells stay alive. Otherwise, it just dies off. With cancer cells, it bypasses that signal. It doesn't care what the others say. It gives itself its own cue. It releases chemicals into the environment that tells itself, keep growing, keep growing. That's a little thing. It doesn't get any encouragement from the outside. It just keeps telling itself, grow, grow, grow. Doesn't matter if there are boundaries. Normally, other cells will say, hey man, this is my space. And they shut off that survival cue, or the proliferation cue. And says, don't grow here anymore. The cancer cells totally go around that, and they tell themselves, screw you, I'm going to proliferate, I'm going to live, I'm not going to do what you tell me to. It just keeps growing and growing and growing. So self-renewal, they do everything for themselves, which to me seems like a subcategory of autonomy, but yeah, they're self-renewing. They just keep replicating over and over and over again. The oldest cancer cells we have isolated were the HeLa cells from early 50s, like 52 or 53. But a woman came in, she had now I'm having a brain fart, it was ovarian or uterine cancer. They extracted the cells from her, they petri dished them, and they kept growing them over and over. She died back in the late 50s, but her cancer cells are still growing and look like brand new cells today. So, did they? Henrietta Lacks?
Yeah.
Yeah. So, in science, you can't use their name until after they're gone and their families give permission and stuff like that. But yeah, that was an interesting story. Her cells are still, they're immortal. And that's what self-renewing means. They'll live forever. As long as they have food and nourishment, they'll just keep growing. So those are the three main characteristics of cancer cells. All right, so identifying them. How we identify them without even having to take them out of the body. We have these things called tumor markers. So a tumor marker, when a cell changes its genetics, it can also change what's on the surface. Like, for instance, when we talked about the E-cadherins, the things that make it isolate or stuck to its neighbors, it will actually eat its own E-cadherins. They're gone completely. There are no structures on the surface, and that's one of the ways we identify this type of cell. So if we see an epithelial-like cell and it has no E-cadherins to hold its other cells, it's a tumor cell. It's a cancer cell. Some of the main characteristics are things like hormones and enzymes. Hormones. If you have a cancer, actually we'll say tumor, if you have a tumor that's growing in your adrenal gland, in the adrenal medulla, what hormone might you expect to overproduce? Epinephrine, yeah. So one symptom of these people are they're going to be like always on the edge, right? But if you took a blood sample on a normal healthy day in the morning, in the afternoon, and the evening, and they had epinephrine high all those times, you might start looking at the adrenal gland. So hormones could do it. What about, I almost said, what about the thyroid gland? What if you had a tumor there? What would it produce? But that's kind of a duh, right? It's going to make lots of thyroid hormones. So people that make too much thyroid hormone, if they have high levels and they look, they may have an actual tumor growing in the thyroid. Cancers themselves are usually more destructive, and you have a decrease in the hormones, where tumors are usually more upregulating the hormones. And we'll talk about that in a minute. Certain enzymes. So there's certain enzymes like CEA, it's carcinoembryonic antigen. And you can look for this as a marker. This is not a normal marker. When this cell turns into a cancer, sometimes you'll act like, I don't know what that was it a sneeze or a cough? Less you could read, type, dig out, I don't know how to say it. But anyway, it can pop this special marker up on the surface so they can help attach to other cells so it can stay alive. But not attach as in bind and get tied down, but attach so it can get nutrients, it can get more stimuli from those other cells. So certain characteristics like enzymes or what we call antigens on the surface, those are all markers. The prostate. Prostate releases an antigen. It's called PSA, a prostate-specific antigen. And if the levels are high in the body, that's usually indicating that they have a prostate tumor. So we can take a blood sample and look for any of these different signs. Too high hormones, too low hormones. Special enzymes that aren't normally released in the blood, special antigens. White blood cells and leukemia, you might see tons and tons of antibodies, abnormal antibodies. So usually these chemicals are things called glycoproteins. We'll talk about that again later. But glycoproteins are sugar proteins. They can sit on the surface of the cell as an identifier. They're like a fingerprint for the cell. When a cancer cell changes, it changes its fingerprint. It may change its fingerprint so much so that it doesn't look human anymore. Then would that be a good or a bad thing? Question. That was a sneeze. Okay. It's bad because it's a cancer cell, but if your body says, you're not me, what's it going to do? Attack it. So you may have had cancer cells growing in your body before, or tumor cells, and your body attacked it. If it changes the fingerprint to recognize it's not you anymore, that's a good thing. Sometimes it camouflages just enough so it looks like you, but it has a little abnormality to help it survive. All these different characteristics are called tumor markers. Some of the benefits of tumor markers is we can actually use those to attack the tumor. So if we can identify what's on the surface of that tumor, we can give you something that goes in your system looking for that specific marker and attack those skills. So here's just a couple. You don't have to memorize these, just up here for the sake of example. Like CEA, that I was talking about before. Would CEA be a good marker that says this is definitely a gastric cancer? No, because if you look above, breast cancer is listed. If you look below, pancreatic cancer is listed, colon cancer is listed. But it's a good marker saying that there's a likelihood that this person has a what growing in them? A tumor. And then we can take this, like the CEA, and then say, okay, it looks like you have a tumor or a cancer growing, and then you can look for any of the other markers that help identify it. So if we were looking at gastric and pancreatic, would SPN be a good one that identifies as definitely gastric? Nope, it's also pancreatic. But if you look at some of the other characteristics, you look at the overall. Like if they have CEA, SPN, and then there's SP439, there's in both of these, but it's not in gastric. So if they have all three of them, then it's likely pancreatic cancer. The next thing you're going to do is start looking at it. So it's kind of a neat thing we can do, where we take a blood sample and start isolating for these specific tumor markers and identifying what's inside of them so we know where to proceed. And that's the significance of the tumor markers you need to know. And I guess I kind of spoiled that one, but so what they're used for, screening and identifying people that are high risk for cancer or have an increased risk, like PSA is a common one. Men get it checked regularly after the age of 40 so that we know, are they increasing the size of their prostate, or is there a problem where it's becoming tumor-like or cancer-like. So identifying, diagnosing specific cancers, and then we can actually watch the progression. So you can keep looking at samples down the road and see how it's progressing. So if they have high PSA and then you do a surgery and their PSA goes down, you know it's a good sign. If the PSA stays high, then you know you didn't do the proper treatment. Get after it. So we can target it, like in the pictures here. You can take whatever that tumor is. If it has a special marker, we can target that marker and attack that marker and destroy it. We can pull a sample of their blood out, we can look for it, and I'm not going to go through all the electrophoresis and all that stuff, but you can look at microarrays and they can target and say, hey, this person has none, this person has a lot of it, so it's more likely to have this kind of tumor. So diagnosis, identification, and progression of the tumors. Then the big one, why can't we cure everything? So if we know all these tumor markers, why can't we just cure these things when we know about them? It's because the tumor markers, when we talked last week, to make a protein, the protein can be hundreds of amino acids long. It can still be the same protein if you just change one amino acid sometimes. It still might work. The problem is that if you have this one protein that's a signifier or a cause of cancer, if you change one amino acid and you try and treat for the old version, that new version can't be attacked. So it's very evasive. It's constantly changing, constantly mutating. 3 billion base pairs, and all it takes is a couple changes to cause cancer. Okay, so building on what we talked about last week, the genetics of cancer. Cancer is a genetic disease. Okay? The whole reason for cancer is some kind of mutagenesis. So it's changing the genetics. Are your genes a definite, if you have bad genes, does it definitely mean you're going to get cancer? No. People get the bad genes for breast cancer and they live their whole life without breast cancer. The genes are just part of it. It's genes plus lifestyle. A lot of studies that they've gone back and looked at cancers, they've looked at Japanese women actually have lower risk for breast cancer. But when you bring them to the United States, even though they were born in Japan, you bring them to the United States and you expose them to our environmental factors, it raises their risk of breast cancer. It's one of those fun things. So, proud to be an American. But yeah, even though their genetics have that ancestry just by putting them in a changed environment. So nature versus nurture. If you're going to be down, you better be tough. So if you're born with bad genes, hopefully you're smart enough to avoid the environmental factors. I love, this guy doesn't do the army justice. You know? He's brilliant. I was watching some show last week. You know what the the animal in America is the #1 killer of people in America? The animal that does the most killing. So, endogenous animal. Deer. Yeah. Deer. Car accidents. Hunters being trampled. I saw a video a long time ago of a guy that put deer scent on himself. Anybody ever seen that? And the deer went crazy and just beat the snot out of the guy. That was definitely nurture gone awry. That guy was Darwin Awards. And then cancer schmancer. No, this isn't my grandmother, but man, it reminds me of her all the time. So my grandmother was smoked from the time she was, I don't know, like 12 or 13. She used to tell me all the time, and then she passed away in her late 80s. So, yeah, she went all the way through cancer schmancer, smoking all the way. But, and that's kind of the thing. People are like, oh, cancer, whatever, and they don't care. Cancer's kind of like the lottery. There are a lot of smokers out there that never get cancer. But the problem is that cancer isn't the only problem. You have to think about heart disease, GI tract problems, bloodborne problems, all kinds of other problems that are happening. Neurotransmitter levels in the brain, they're all getting affected regardless of whether they get cancer. Since we're talking about cancer, let's stick to that one. Moral of the story is don't smoke. If you haven't heard, it's bad. All right, so cancer genes can be inherited, introduced, or due to mutations. So the primary one we're going to look at is the key mutation, lots of changes. You can have a flaw that your parents gave to you, which you should thank them every day if that happened. But anyway, you can get things passed down from one generation to the next. Typically, cancers affect the generation. They affect somatic cells, not what cells? Somatic are your body cells. What are the G words? Not bacteria, but germs. Nobody's ever heard that word. So, typically, these mutations don't affect germ cells. Typically, mutations affect somatic cells, which are in the person themselves. The germ cells will be the egg and the sperm that pass it down, and something usually happens after that. So, mutations. Right? If they're introduced, it could be not because of a mutation, but because like a virus came in. What's a virus do? It tries to plant its DNA or RNA inside of you to change your DNA. So it can be introduced by something else. And we're going to talk about these terms, proto-oncogen and oncogens, over the next several slides, so you don't have to write it down now. But cancers are always a change in the genes. So here's an example. So Burkitt's lymphoma. Burkitt's lymphoma can actually be introduced by lots of other things. So it can be introduced by HIV, malaria. So malaria, we're talking about an infection because of a parasite. HIV is because of a what? What is HIV? The V tells you it's a virus, right? And an Epstein-Barr virus. Anybody know what Epstein-Barr virus usually causes? Mono. We'll talk about that a lot. But these viruses, they try and get in and they try and change the cells. H. pylori, what is that? That's not a virus. That's a bacteria in your stomach. All of these things can get in, they can cause changes. And any of them can actually lead to things called Burkitt's lymphoma. So, Burkitt's lymphoma, would that be a benign or malignant tumor? Malignant, how do you know? Because it's a lymphoma. So, leukemias and lymphomas are always malignant. They're always dangerous. And what they do is they get in and they affect these things called proto-oncogens. Proto-oncogens are good. Proto-oncogens are genes that do good things. They make good enzymes or make good factors that help cell growth. Regulate cell growth. Once somebody gets Burkitt's lymphoma, it comes in, it attacks the chromosome, changes it, and these genes get formed into something called oncogenes. What do you call a person that studies cancers? An oncologist, right? So oncogenes are gene or cancer-related genes. And then these lymphomas are typically because of proto-oncogens getting turned into oncogens. So in this situation, this lymphoma, look at this. You know what these little red things are in this picture? They're concave-shaped cells with no nucleus. The red blood cells. So all of these purple-like ones are white blood cells. What do you know about the proportion that's weird? They're almost as many white blood cells as there are red blood cells. It shouldn't be like that. You should have barely any white blood cells in a whole field of red blood cells. So in this situation, they have overly expressed genes that make the white blood cells rapidly replicate. The bad thing, you think, well, if they have tons and tons of white blood cells, they should be really healthy because anything gets in, they should be able to fight it, right? The problem is that when you mass produce something or overproduce it, what happens to the quality? It starts going down. So the quality of these white blood cells go down. They don't work like a normal, healthy white blood cell. We'll talk about Burkitt's a little bit later, but for now, you just want to remember Burkitt's lymphoma, is it malignant or is it benign? It's malignant because it's a lymphoma, and then it's because of these proto-oncogens that we're going to talk about in just a little bit. Okay, here's an example of inherited. So that was introduced, the last one was introduced. This one's inherited. You can get a bad gene, and we know where the bad genes are. So they're called BRCA genes, and primarily you're going to hear about BRCA1, BRCA2, and you can see the locations of these different genes. We know the chromosomes. We know the, what do you call the L word for the location? The locus, yep. And we even know that the alleles have changed here. So we know all about this. But the problem is that it's in your genetics. Is it easy to change this? Why? Because these are your genes. They're in how many of your cells in your body? Almost all of them, except for red blood cells, right? With the exception of it. But yeah, they're like every cell of your body except the red blood cells. So if you had to change them, you have to change every one of them. These are usually causing effect in a tumor suppressor gene. Guess what a tumor suppressor gene does? Suppresses tumors. So I always see these things as being the brakes. If a cell has a malformation, this tumor suppressor gene comes on and goes, and it stops it from replicating. Guess what happened to the brakes in this type of cancer? You broke them. So what's the cell able to do now? Rapidly replicate. If the tumor suppressor gene kept it from replicating, the tumor suppressor genes usually recognize that this is a bad, dangerous cell. We have to kill it. But this cell is going, I don't want to die. And then if it breaks the tumor suppressor gene, that brake is released, and that cell goes, woohoo! Hits the accelerator and starts replicating over and over and over. So the tumor suppressor genes usually stop proliferation and stop growth and replication. Now it's just going crazy, and it's replicating like crazy. The other thing we'll talk about later is repair machinery. These also help with repairing the DNA. They hit the brakes and they start helping to fix the DNA. If these are broken, not only do you have acceleration, you're replicating rapidly, but what's going on with the DNA? It's going to stay broken, right? It's going to stay flawed. So what do you know about every copy that cell makes? It's just as flawed as the original one. And then we already answered this question, but does a bad gene ensure cancer? No. Nope. In fact, good genes don't ensure that you're not going to get cancer either. So there's a special feature called epigenetic change. Epigenetics are relatively a new science. Epigenetics are looking at your genes, but what happens to your genes after you're born? This is weird. You can have a normal copy of a gene and a bad copy of a gene. And based on epigenetics can say, well, we're going to turn off this good copy. Now what do you have left? The bad one. Or epigenetics can do the other way around too. Even just genetic disorders, and I always think about putting this in the slides even though it's not in your textbook. There's this disorder that's called Angelman's syndrome, where the kids are born really short, they're mentally challenged, but they're always really happy-looking children. They're really skinny, little short little kids. We know exactly where the gene is that causes Angelman's. But if you take the same person with the same genetics and the same flawed genes, they may have something called Prader-Willi, which they're tall kids, they're extremely chubby, but they're still happy. They have similar characteristics as far as the happiness, but all the other physical characteristics are very different. They're also mentally retarded too. But you have the exact same gene. If you take the chromosomes from both of these people and you look, that same gene is exactly flawed. They think that something happens to your genes after you're born, and while you're just living normal day-to-day life, it causes a change in expression. Same disorder, but basically different expression. So you get two different names. It's called epigenetics. And I'm not going to go super deep into the epigenetics. I put a couple slides in here so that if you want to look at it, you can. It's basically saying that your genes, once you have them, the way that they're coiled, the way that they're unraveled to turn them on, can be changed just by your environment. So something as simple as smoking cigarettes, the toxins can get in and change the way these genes are turned on. Eating the wrong thing in your food can get in, bind to these genes, and affect the way they're turned on. If we got time, we'll talk about that a little bit, like charring meat on the grill. Delicious, but bad for you, because it has a lot of epigenetic influence. So, like I said, the extent of epigenetics is pretty much isolated here. It's a new, relatively new science. And your book doesn't talk very much about it at all, so I'm not going to talk much about it. But it's just saying that you can have good genes, you can have bad genes, and things can change. Right? So how do mutations work? The next type, and this is the primary type. So mutations, of course, they affect the DNA. What was a point mutation, do you remember? It's affecting one base pair, right? So it's going straight to a base pair or one part of a gene and changing that. There are two types you could have. One was where you completely eliminate and it changes the whole frame. What was that called? Frame shift. I didn't know how to word that. So frame shift. The other one is where you can take one letter and replace it with another one. That's called substitution. So frame shift is where you delete a whole letter or you add a whole letter that doesn't belong there, and it shifts the whole frame in the reading. the point mutation, that's which one's better? The substitution for the frame shift. Substitution. And the substitution where you just switch a letter. So if you're lucky, remember you can have one amino acid that has potentially 6 different codes. Change one letter, you might get lucky and keep the same amino acid. So mutations get in there and they can either cause point mutations, or they can actually cause chromosome aberrations or alterations, where they break an entire section of chromosomes. Then we talked about that. You can have the translocations where you plant it somewhere else. You can have inversions where you flip it in and you put it back in the wrong way. You can have complete deletions. So that's what mutations do, whether it's a point mutation or breaking the whole chromosome. And this is kind of a key too. Does cancer usually happen after one mutation? Nope. Cancer is typically known as an accumulation of mutations. That's why, who do we usually associate cancer with? Young kids or elderly? The elderly, the older populations. Because we've been living our life exposed to all these bad things, and they just accumulate, and get worse. So predominantly a disease of aging. And basically the replication goes faster than apoptosis. Yeah, here it is. So I put some examples here. You can see normal tissue, and then you can see the benign polyp, and this change in the structures, and then it gets worse, and now you have a cancer. So, huge change from the original tissue. This next slide is right from the textbook. And this is just one example. We're talking about polyps growing in the colon. So here you have the normal epithelium that's growing along the colon. You get one mutation, we typically know where the first mutation's at. So the cells look pretty normal, they may be growing a little bit weird. Once this mutation happens, then we know the second mutation is going to be on the 12th chromosome. We know exactly where it's at. There's the second mutation. Now they start looking really weird. And typically, if they're an adenoma, what are they growing out of? A gland. Yep. So you start having these glands that start getting these abnormal shapes and abnormal growths. And then we know, watch for the third chromosome change. Here, you're going to have this really big growth protruding out, which we associate with the polyp itself. And it keeps growing and growing. And now we know if we've had these 3 accumulated changes, we can predict the next one. We know exactly where it's at. If you get this one, that's when that that cell becomes invasive. It turns on genes that activate enzymes that cause it to eat the other neighbors and move into the tissues. We know the progression of each of these. So if somebody's getting, in their 40s, they're getting a check and they see a mild polyp and they see this change here, they want to try and catch it while it's this change to prevent it from going here. So they'll biopsy, they'll look and see, is this the only change? If it's this, what should they be worried about? So they have all 3 of these changes. They biopsy the tumor. Biopsy means take a piece of tissue out. And they see in the genetics that this has changed. What do they know about that? Is it likely to be benign or malignant? Malignant, yeah. So they know that it's probably deeper in the tissue. They start looking around that person's body because it probably moved into the lymph or the vessels, blood vessels, and moved. So we know the specific progression of these. It's just hard to treat it. Number 1 is because it's hard to see early on. Number 2 is because we don't know all of the changes that are happening in addition to these. And these aren't...
So there's no marker in this, like when we do blood analysis, there's no that doesn't automatically trigger like an increase in white blood cells or...
No, not automatically. Yeah, that's a good question. Because if these are normal cells that still have the same fingerprint to say they're you, should it trigger an increase in white blood cells? Yeah, it has to have some kind of change in special tumor markers. Like we showed earlier, that CEA has a sensitivity change when we're talking about gastric ulcers. So you might see a change in the CEA level in their blood and say, hey, we need to look for a cancer. We don't know yet if it's in the stomach or if it's in the GI tract or whatever. But in this situation, that's why after about 40, you start getting all these, you have to start getting checkups regularly because cancer starts replicating because you've mutated so many times for that time. Sun exposure, X-ray exposure, just toxins in the environment. You don't have to memorize this. I'm just giving you an idea. That you can see a lot of these markers. We know exactly what's going on. We know the genes even that are being changed. The problem is that it's really hard to isolate these. So it's really hard to stop it. We know the progression, we know the change, but it's hard to stop it. And actually, I'm going to stop here. We'll take the break and come back in 5 minutes, just so we have a nice clean break.
you would